All right, so I just want to go over our notes for the DNA section. Just to give you a heads up, you can go to your workspace. You will have a new workspace available to you called DNA. Underneath there, you go to the notes, and here's a pair deck, and I'll also probably put in a Quizlet cards here pretty soon. But we'll go ahead and open up this DNA PDF here, and we'll go through it. Big thing that you need to understand is there's a big difference between DNA and RNA. We're first going to talk about DNA, which resides inside of your nucleus. There's 6 major features of DNA that I need you to understand. It has 2 main sides. And those sides are made up of 2 different chemicals, what we call an alternating sugar and phosphate group. Then it also has what we call a nitrogen base that form, if we refer to it as a ladder, the sides would be the sugar and phosphate groups. And they're held together by the hydrogen bonds like it says there on the page. And then the nitrogen bases that kind of fit together in the middle would be the ladder rungs that you would climb up. We have 4 of those nitrogen bases in DNA, and those 4 bases join together in certain ways to form the rungs. The 4 are right there. It's adenine and thymine, and then guanine and cytosine. Really, when we talk about this, you just have to remember A goes with T and G goes with C. You're not going to have to recite the names or anything like that. If you get into some further biology courses, you go to college and you take some more advanced biology courses, you probably will be asked to know that. As you can see right here, this is the actual shape of our DNA. It's what we call a double helix. As you can kind of see, these would be your sides, which are the sugar bases. In this case, DNA, it's deoxyribose is the sugar. And the phosphate groups will make up this outer area here. Okay, this line that's on the outside. Then you have the base pairs, again, A to T, adenine to thymine, and guanine to cytosine. And as you can see, here they are structurally. The pyrimidines are a single carbon ring, while the purines are a double carbon ring. And that's kind of how they fit together. In the middle being held together by hydrogen bases there also. So how do we get DNA from the deoxyribonucleic acid? You say, the question is, how do we understand, or why do we think of it that way? Well, if you get down to the microscopic size of it, it starts out in your cell as a chromosome, and then it will kind of unwind into this double helix area that we look at all the way down to the base pairs. So the nucleic portion is related to the part of the cell in which this is chemical is located. So when we say nucleic, we're talking about it's located in the nucleus. The deoxyribo, or the stem for the deoxyribose, which is the sugar molecule. And then, of course, the acid relates to that phosphoric acid that holds that side together. During mitosis and meiosis, we go through a process that we call DNA replication, where we have DNA polymers, which comes in pretty much unzips the DNA molecule, kind of like a zipper here. You can see in step 1, it's just a structure. In step 2, we have it kind of coming apart. And then while that's doing that, we have matching base pairs come together to form 2 new strands, but identical to the original strand of DNA, which allows us to pass our genetics on. So we get 2 or a doubling of that area. That way we can be able to pass on that genetics to the next cell that we're making. When we talk about changes or copying, messed up copies in the DNA messages, we're talking about mutations. Again, I've talked to you guys about this before. We're not talking about like X-Men mutation. We're talking about diseases of some sorts, improper splitting of DNA in meiosis can cause what we call non-disjunction, which means that a chromosome could have an extra pair or not an extra pair, but an extra chromosome. So we would get like what we call trisomy. Trisomy 21 is what causes Down syndrome. That is a mutation. We'll talk a little bit when we get into genetics about sickle cell anemia. That's a mutation in the line of DNA also. So like it says here, this may cause a different trait to appear. It may not. Sometimes there are harmful mutations. Sometimes there are mutations that are not harmful. So that's DNA. Again, DNA is located in the nucleus. It's named due to the sugar, so the deoxyribonucleic or the deoxyribose. Then we move on to what we call RNA. And RNA is a single strand. You can kind of see it only has one of the double helix strands, so it's a single strand, and it carries information in different ways. The two that we're most concerned with is messenger RNA, which is a long strand of RNA nucleotides, and it forms complementary to the strand of DNA. Now, the big difference between the nitrogen bases in DNA and RNA, instead of thymine, we have a base called uracil. So uracil matches up with adenine. So when we talk about RNA transcription, when we are going to transcribe or we are going to write a line of messenger RNA, which happens in the nucleus as the DNA splits, we are going to put in the nitrogen base uracil instead of thymine. So you can kind of see it here, GU or GCU, ACG, GAG, CUU. So instead of T's, where the U's are instead of T's, we have a U for uracil. The next molecule of RNA that I want to look at is called transfer RNA. It is smaller segments of RNA, nucleotides, and it's kind of got this clover leaf shape. And really what we are concerned about is this bottom loop right here called the anticodon loop. Now, codon is a 3 base pair, and that's how we break apart or segment the line of RNA so that we can understand what amino acid is going to be attached to this other end of the transfer RNA, because really that's the job of the transfer RNA, is to bring the amino acids to the ribosomes. So what you would have is you would have 3 complementary bases here that's going to match up with the line of RNA as it goes. Now we have 2 different processes. I've already kind of talked about transcription. The DNA is going to unzip, okay? And the major thing about DNA transcription is we are using the DNA code to transfer to an mRNA strand that comes complementary to the DNA strand, so that we can take that to the ribosome, so then the tRNA can bring an anticodon that has an amino acid tied up to it, because our main goal is to make proteins. Okay, so the steps of DNA transcription are right here. I'm not going to read straight through here, but basically what happens is the DNA unzips much like it would happen in DNA transcription. And what we have is we have a filling of RNA nucleotides based on this RNA polymer. So it just fills in and it builds this line of mRNA. Once it's synthesized, it will leave the nucleus and it will go to where the it'll go through the nuclear pores and then go to the ribosome. Now at the ribosome, we have translation happening. So transcription, I've always explained to as something being written. Okay, so we are writing that mRNA line and we're taking it to the ribosome. Now the tRNA translates that based off the anticodon matching up complementary to the mRNA. So again, we're talking about RNA. We're going to use the bases adenine and uracil and cytosine and guanine. Okay, so these 3, what we call anticodon, has a amino acid that's attached to them. As it comes in, you're going to have another polymers come in and take care of putting those amino acids together in a streak.
Now, we have 20 known amino acids that make up proteins. And it's based off our codons, like I said, which is that 3-base code in DNA and mRNA. Each codon is transcribed into the mRNA code. So, for instance, I'm not sure what all this mess is here, guys, on the notes. I apologize. But you would use this column as your first base, this would be your second base, and this would be your third base. So we'll have some more practice with this, but as you can see, so if I went UUG, this first one, it would be leucine, I think is what I'm reading there. So this would be your first base, this would be your second base, and then you would have a third base matching up over here. So for instance, I gave you AUG. So if we want AUG is the last one, it'd be methionine. So that's how you figure out DNA transcription. We put them together like that into long chains, and what we do is we make proteins that way. So that's basically DNA and RNA. Remember, the differences are DNA has two sides. It has the sugar deoxyribose, and it has the nucleotide base thymine, whereas RNA is a single strand. Its sugar is ribose sugar only, and instead of thymine, it has uracil. So hopefully we are good on those. We're good on those two concepts. We'll do another pair deck. Again, like I said, I will put a Quizlet Live cards up for you to study also.